All right, y'all. So as you worked through the first question on the activity, I hope that you recognized this idea of redundancy, right? So the fact that multiple codons will all result in the same amino acid. And this is really, really important for the idea of redundancy. It makes it less likely for a mistake to be made when proteins are being synthesized, right? So the fact that the first and second positions are by far the most important in creating, in putting the right amino acid onto a growing protein chain. That makes it much more likely to have the right sequence of protein. Okay. This it also lets cells be a lot more efficient when they're making proteins. So the reason for this is pretty straightforward, right? So it means that they only need to make a single tRNA and very, very, very commonly a single amino acyl tRNA synthase for multiple codons. So for example, a single tRNA and a single amino acyl tRNA synthase are used for each of these three codons. Okay, that means the cell can just be more efficient. It doesn't have to use so much energy to make three different tRNAs, three different amino acyl tRNA synthetases, when one can do the job. Because this third position really doesn't matter. It's called the wobble position. Okay, so C and A in the wobble position actually do typically matter. They're almost always partnered with the normal Watson and Crick partner G or U respectively. However, when U or G is present on the anticodon, these will recognize either A or G or U or C, right? So if these are present in the anticodon, it can recognize either of these two via non-traditional Watson and Crick base pairing. In addition, there's this other nucleic acid called inosine, which it's a modified version of adenosine, where the amino group that points up ends up being converted to a carbonyl. And we'll look at the structure of that here in a second. But the idea is that the presence of inosine on these tRNAs will recognize either of these three nucleic acids on the on the codon. So it just makes things more efficient. Okay. Let's see. Okay, so here we go. So this just shows you how this non-standard nucleic acid, inosine, is able to base pair with three different nucleic acids, either uracil, cytosine, or adenosine. So inosine is shown here in pink. Hopefully you notice it looks an awful lot like adenosine, except this NH2 group is converted to a carbonyl, which changed the base pair pattern, right? So adenosine is a donor at this position, an acceptor at this position, where inosine becomes an acceptor and a donor, which makes inosine quite able to base pair with either uracil or cytosine, right? So uracil, cytosine, or adenosine. So let's continue on and think now really critically about our different RNA molecules. And now we're going to focus on the tRNAs. Okay, so the tRNA, we said before, is the adapter. It is the key adapter that allows the correct anticodon to be partnered with the correct amino acid. And it's this position right here at the 3' end of the tRNA where the amino acid gets attached to. Okay, it consists of 4 stems. Okay, the 3' and 5' ends, which are shown right here, are both part of what are called the acceptor stem. Okay, it's interesting and notable to point out that this stem is not base paired all the way through. There are 4 nucleic acids on the 3' end that are not part of a base pair in the stem. Okay, But there are 7 nucleic acids here that base pair that form a really important A-form helix in this structure. Okay, so that's our acceptor stem. The D arm shown here. So it is 4 base pairs with this big loop at the end. And it's called the D arm because of the presence of these 2 modified nucleic acids. The D stands for dihydrouridine. So if you draw out uracil or thymine, right, remember that I won't draw the whole thing out. Oh, I guess I will. So I'll stop here. So remember there's a double bond here. That's really important in making the structure planar. But dihydro has that reduced, and those carbons are sp3 hybridized, making this base non-planar, presenting a situation where it's not good at pi stacking, and that is actually really important. Okay, so the D arm is named because of the presence of dihydrouridine. We then have a 5 base pair stem. So this is our anticodon loop. It's 5 base pairs, and then this kind of large loop at the end of it that contains the anticodon. Okay. I point out here that so the anticodon is going in the 5' to 3' direction here, right? So here's 5', here's 3'. So 5' to 3' is going in this direction. That's notable because it'll partner with the codon in the opposite direction. So it will partner with the codon G, C, and then remember inosine will partner with A or C or U. So this will partner with GC and then either A, C, or U. The last part of it is called the psi arm. Okay, it's noted because of the presence of a thymine here, which is interesting. This is called pseudouridine. So the psi stands for pseudouridine, and we'll look at the structure here shortly, but it's a modified uridine where the attachment to the ribose is oriented differently. Okay, so this loop is characterized by this T psi C sequence. Okay, and then it always ends at the 3' end with a CCA. Okay, that's important because the aminoacyl tRNA synthetases, the enzymes that put the amino acid on right here, are looking for that sequence. Okay, and it has to be a not based paired sequence. So this is an important feature at the end of every single tRNA. All right, y'all, so this brings us to a point where we want to kind of explore and think about the structure of tRNA and the reason why there's so many modified bases. As we've mentioned, tRNAs are the adapter molecules that put the right amino acid, that pair the right amino acid with the correct anticodon. Okay, they tend to be between 73 and 93 nucleic acids in length, and the reason for this is the presence of a variable loop, which we'll see here in a second. Overall, as we saw in the previous slide, it forms a cloverleaf structure when we're thinking only about the secondary structure. This structure is formed by the presence of these stem loops, right? There are 4 stems and 3 loops. We have the acceptor stem up here where the 5' and the 3' ends are. It's also where the amino acid is attached to. We have our anticodon loop down here, and that's where the anticodon is. We have our D loop, or sometimes called the DHU loop. Okay, that's where we have the presence of multiple dihydrouridines. And then over here we have our T psi C loop. These things are absolutely loaded with modified bases. Okay, and those modified bases are really important when we think about the overall 3-dimensional or tertiary structure. Okay, so if we now consider the tertiary structure, it's not a cloverleaf, it's a hook or an L. Okay, there are 2 continuous helices, right? So we have one continuous helix here, which is terminated by the 3' end, and we have one continuous helix here, which is terminated by the anticodon. Okay, this is important and relevant, right? And the presence of these two helices in this tertiary structure is made possible by modified bases being deliberately incorporated into positions to impact and prevent Watson and Crick base pairing and pi stacking. Okay, so here we have the structure of all of the modified bases common in tRNA molecules. We talked about pseudouridine and dihydrouridine. Both of them are uracil derivatives. Okay, so dihydrouridine is different because it's a reduced form of uridine that does not have a planar structure. So these two carbons are sp3 hybridized, not planar. Pseudouridine actually has the ribose attached at a different point. Normally, the ribose is attached to this nitrogen. It's rotated so that ribose is attached to this carbon. As you can imagine, that will impact the way that the Watson and Crick face is presented to a helix. Okay, guanine derivatives. M7G, this means there's a methyl group at position 7, okay, right there. This is not the Watson and Crick face. This is the Watson and Crick face of guanine, so this actually impacts what's called the Hoogsteen face. It's the face that is over here, right? So the Hoogsteen face uses this nitrogen and this oxygen as hydrogen bonding partners. So this impacts hydrogen bonding on the Hoogsteen face. M22G, this has two methyl groups on this nitrogen of guanine on the Watson and Crick face. M1G has a methyl group here, again on the Watson and Crick face. And the adenosine derivatives, we can have a methyl group at this position, or we have inosine, which we've already talked about, where we convert this amino group to a carbonyl. So you can certainly imagine how these can impact pi stacking in some cases and hydrogen bonding in some cases. Okay, all of that's really relevant and important because they are incredibly important in maintaining the three-dimensional structure of our tRNAs. Okay, so what this is kind of hoping to try to point out is some of the non-standard base pairs that occur in this hook structure of tRNA. Okay, so note here that there are two continuous A-form helices, right? One of them is right here. It's formed by the acceptor stem and the T-psi-C stem. Okay, one continuous stem here. And then there's one continuous helix here formed by the anticodon loop and by the D loop, the dihydrouridine loop. Okay, and this helical junction where there's this sharp turn, that is maintained by these non-standard nucleic acid hydrogen bonding, right? And what you'll notice here is that lots of these that are absolutely conserved are formed by some of our non-standard nucleic acids. So our pseudouridine, which is incredibly important and absolutely conserved, makes a critical hydrogen bond with guanine on the D loop. And that is really important in maintaining the status of this helical junction, making sure that we have this sharp turn, which gives tRNAs their L shape. Okay, and other of these these nucleic acids play important roles as well. We won't go into all of them. But what I really wanted to point out is the role of these non-standard nucleic acids. They make sure that we can't form a constant, stable, continuous helix. We have to have some sort of other structure, and that other structure, this hook, this shift, like this curve, is stabilized in many ways by non-standard hydrogen bonding. The other important thing to notice here in the structure is at the end of the anticodon stem, our codons here are all flipped out, right? That means they are not part of hydrogen bonds. They in this structure, they're not hydrogen bonding with anything. They're flipped out, And that's really important because we know that they need to make contact with the mRNA in the ribosome, but they also need to be able to contact specific amino acids in our aminoacyl tRNA synthetases when we try to load an amino acid onto the 3' end of our tRNA. Okay, so these are looped out. Okay, and this specific anticodon loop we see here, right? Our anticodon is AG, which would code for a U, and then something else. So this would either code for a phenylalanine or a leucine. In this case, it's a G, so it codes for the leucine. So at this point, why don't you go ahead and take a few minutes to work through the next couple questions in your activity, which explore the structure of these non-standard nucleic acids and also think about the interaction between the codons and the anticodons of tRNA.